welcome everybody. Thank you so much for joining us today at the Diagnostic Landscape for COVID-19 virtual meeting. This is the first time that we are doing this meeting, and it's the first of our 4-part Together as One COVID-19 series, which covers diagnostics, vaccines, therapeutics, and post-pandemic insights. And I'm Emily Lee. I'm the founder and CEO of BioInsider. Our goals with this COVID-19 series are to bring the scientific community together while giving back in this challenging time, as COVID really turns our lives upside down. And we are donating our net proceeds to the COVID-19 Solidarity Response Fund for the World Health Organization to help fight COVID. And we want to thank our sponsors, Sinobiologics, LabCorp, AgroBiosystem, BioFire, and CW Bio for helping us make this meeting happen. A big thank you to all of our speakers, moderators, and all of you for being here today. And I would like to thank the BioInsider team members for helping me in the last few weeks to launch this meeting. In such a short amount of time, guys, we've been able to pull it off in just over a month. So as you might already know, BioInsider is a startup company. We connect experts and decision makers in the scientific community to enrich knowledge, foster collaborations, drive the success of their research, and spark innovations. So we focus on interaction among speakers and attendees, and we're happy to bring in the speed networking and the breakout discussions today at our meetings to close this gap by providing an intimate setting for you to connect with people. And of course, these are all in addition to our critical scientific presentations. So what I consider to make this meeting successful today is if you learn something new today from our speakers and your fellow attendees, or you make a new connection and you will follow up via LinkedIn or emails, which helps with your current project, generate new ideas, or a potential collaboration. That's what I consider is going to be a successful meeting. And so before we start the meeting, just want to go over a few housekeeping items. Throughout the presentation, you can send the questions to me, to the host only. So Emily Lee, if you hover to the chat box and choose the option of sending your questions to the host, that will be answered during the Q&A session. Any unanswered questions today will be answered by our speakers and emailed to you next week. We will email you the on-demand recording of the meeting after the meeting. So for the speed networking, guys, turn on your video and unmute yourself so that you can connect with others and interact with them, okay? So with no further ado, I would like to introduce our chairperson, Dr. Carl Volkeding. He is the professor of pathology at the University of Utah and a medical director of genomics at bioinformatics at the ARUP laboratories. And Dr. Volkeding has a long-standing involvement with in the translation of new technologies into molecular diagnostics. And this interest has focused over the past several years on next-generation sequencing. And he's currently the chair of the College of American Pathologists Genomic Medicine Resource Committee. And it's my honor to work with Carl over the last few years for other conferences that I've worked on. And I could not find a better chairperson than Carl for this meeting. So Carl, please take it away.
Well, good morning, everyone. I'd like to welcome you to our meeting today, the Diagnostic Landscape for COVID-19. We have an outstanding series of speakers that it is my privilege to be able to introduce today. I just want to give a few introductory comments before I introduce our first speaker. This is certainly a remarkable time in our global history together. We, as human beings, are subject to be infected by at least 7 known coronaviruses, 4 of which cause very mild respiratory symptoms. And then in 2003, the sudden acute respiratory syndrome virus, the original SARS virus, was identified in 2003, followed in 2012 by the Middle Eastern respiratory syndrome virus, or the MERS virus. Again, both coronaviruses. And now in 2019, we have SARS-CoV-2, the focus of our meeting today. Interestingly, the origin of these viruses, their genomic similarities are substantial to coronaviruses that are harbored in bats. And also the SARS-CoV-2 virus has sequences of high similarity to pangolins. And so therefore the current theory is that the SARS-CoV-2 virus is essentially a zoonotic transmission from an animal host into human. The first report of the SARS-CoV-2 virus, as many of you know, was reported in December of 2019 from the Wuhan region in China. And from that initial report in December of 2019, by January 26th, 2020, so just a little over a month later, in China, there had been approximately 2,700 laboratory confirmed cases of SARS-CoV-2 and 80 deaths. Since then, there's been a remarkable proliferation and global transmission of the SARS-CoV-2 virus. As of yesterday, on the Johns Hopkins COVID-19 dashboard, some statistics of interest. There are over 8.2 million reported cases of COVID-19 globally across 188 countries and territories. In the United States, and in that context, there's been over 445,000 deaths reported and greater than 4 million reports of recoveries. In the United States specifically, we now have over 2.1 million confirmed cases and just over 117,000 deaths. Worldwide, the virus is continuing to proliferate. And even though some countries are seeing declines in their cases, other countries are seeing increases. So we are still very much in the first wave, if you would like to term it that way, of the SARS-CoV-2 replicative infection and transmission globally. This virus, like the original SARS virus, binds to the angiotensin converting enzyme 2 receptor in human tissues, and that receptor, the ACE2 receptor, is expressed on many different tissues in the human body. It's essentially the cellular doorway for the virus to enter cells, replicate, and then the host immune responds to that, which causes serious complications, including fatalities. So today we're going to really focus on the diagnostics of this virus, which have been essential for understanding both the diagnosis, the transmission, and the proliferation of this virus, both molecular and serologic approaches. And now I'd like to turn over to introduce our first speaker, which is Dr. Timothy O'Leary. Dr. O'Leary, who's based in Maryland here in the United States, is currently an adjunct professor at the University of Maryland. He served as the Chief Research and Development Officer at the Department of Veteran Affairs from 2013 to 2015. He has a distinguished and stellar career. For over 15 years, he chaired the Department of Cellular Pathology and Genetics at the prestigious Armed Forces Institute of Pathology. He is a past president of the Association for Molecular Pathology, and he served as editor-in-chief for many years for the Journal of Molecular Diagnostics. And Dr. O'Leary is going to present today, the title of his talk being Comparing Diagnostic Methods for COVID-19, Practical and Statistical Considerations. Dr. O'Leary, welcome to our meeting, and thank you so much. I will turn the podium over to you.
Okay. Okay, I presume people can hear me. We're going to talk about some things that are really pretty common to all laboratory tests, which give dichotomous outcomes, present, absent, which is generally the first thing we're looking for SARS-CoV-2 infection. And been asked many times, people are asking this question, which is better? What's the best test for SARS-CoV-2? Of course, there is no good answer to that. I'll be talking a little bit about statistical considerations, and I will play off a comment by Richard Hamming and suggest that the purpose of statistics is understanding, not p-values. Sometimes we seem to get lost in that. And of course, understanding laboratory tests and the evaluation of laboratory tests requires that we understand the bias associated with that evaluation. I would suggest that there is no unbiased evaluation of a laboratory test that's ever been reported in the literature. So the question is understanding what those biases are and then trying to make sense of those biases in the context of what we're doing. There are lots of different kinds of bias: selection bias, spectrum bias, verification bias, test evaluation bias. We won't talk about all of those, but we'll comment on a couple of them. And the most important, I think, in this case is spectrum bias, a form of selection bias, where we evaluate a test or a test strategy in a way that isn't representative of those to whom the test will be applied. For example, we choose our COVID-19 positive specimens from people who have, florid COVID-19 symptoms and signs. We choose our negative specimens from people who've never been exposed. And this, of course, is not usually the group of people that we're considering. Yes, we are certainly going to see some floridly infected people, but a lot of people that we come to see are showing minimal signs. Spectrum bias may also show up in the question of when we are getting our specimens. Our highest positivity rate, no matter which site we look at, almost, for COVID-19 is found in the first week of infection. So if we are applying that test in people that are in the third week post-onset of symptoms, we can't expect the same results that we saw at the beginning. And spectrum bias has been built in to many of the evaluations that we see in the literature. Very commonly, we'll see that one site has been tested early on in the course of disease, and then in the course of deciding to do a study, people have looked at the same patients a week and a half later. We also have problems of verification bias. And a good example would be to use people who never had any symptoms that were at all like COVID-19 is our negative control. This is frequently done. Often it's the best thing we can do, but we should recognize the biases in our evaluations. And finally, and I think this one is the most important for COVID-19, imperfect reference bias. Imperfect gold standard is used. Outcome measures such as sensitivity and specificity will be biased and will favor the gold standard. Consider this, for example, in the evaluation of swabs. We'll come back to this in a minute. If we evaluate swabs and we have decided that the perfect swab is going to be the nasopharyngeal swab, then nothing can look any better than that. You could match it in theory, but you could in fact have something that was better than the standard nasopharyngeal swab, and you'll be classifying it as a false positive, which kind of doesn't make a whole lot of sense in the context of RT-PCR testing for SARS-CoV-2. Finally, and we see this appearing again and again and again in the literature, discrepant analysis. It's never a good idea. We're comparing A and B. When they agree, we assume without further evidence that both A and B are correct. When they disagree, we use a third test, C, as a resolver. The problem is that this is biased. And sometimes if you do C, it's not going to agree with either A or B. And the result of using discrepant analysis is that the performance characteristics of the test that you are originally using are always artificially inflated. There is always bias associated with discrepant analysis, and it should probably never be employed. Incorporation bias. We employ one of the procedures as part of the gold standard. This is fine if you're looking for substantial equivalence, and it's something that's actually occasionally done in FDA type evaluations, but it's not capable of identifying superiority. So how do we avoid these fails? Well, we start by asking the question carefully. You know, what are the sensitivity and specificity of A and B? Does test A and B really have different performance characteristics? Is the sensitivity of B non-inferior to that of test A to within a tolerance? For instance, is it within 10%? Or is test B superior to test A? Then you need to define carefully how you'll answer the question, and you should define these questions and the way to answer them before you actually start the study, rather than gathering up a whole bunch of cases that because you've been doing diagnoses and making comparisons post hoc. You should decide what's a clinically meaningful difference. You know, I'll consider my test to be good enough if it's at least as sensitive as the Abbott ID Now test. Or I'll consider my test to be good enough if it's within 10% non-inferiority margin of the CDC test. And then decide, is this making any sense? Do you really care if the sensitivity and specificity of a test are 95, 99, or is test B just as good if it's 93, 95, and it costs a tenth the amount to perform? You want to be able to guess what your sensitivity and specificity of the test is likely to be. And, you know, sort of jumping to the end, when it's all over, don't fall into the p-value trap. Don't conclude that a 10,000 patient study that shows a 1% difference is better for helping you choose A or B than a study that shows a 30% difference at p equals 0.005. How much of a wrong conclusion are you willing to accept? Type 1 error, finding a difference where there is none, or type 2 error, failing to find a difference that's really there. Most studies I see in pathology reported in all of the journals, and this includes the microjournals, accept the default value of p equals 0.05 for alpha. Few studies seem to pay any attention at all to beta. Nonetheless, people will then conclude, because they find no difference, that there is no difference between A and B. And of course, this is a false conclusion. So you really want to define what that error tolerance is, ideally. You would like to know not only the type 1 error, but the type 2 error. And the type 2 error is really falls into the power. If you take 1 minus that type 2 error, we get something called beta, or the power of the test. So if we have no more than a 15% probability of failing to find a test non-inferior, for example, then we say it's powered at 85%. Finally, before doing the study, before gathering the data, you really ought to be determining what the statistical testing method is. You know, you may decide you're going to compare your standard against a reference material and then conduct McNamara's test. Or you may decide that you're going to conduct Little's test. And these would be the two most common tests if you are actually going to try to power a study adequately. We'll talk about how difficult that is. So again, what does power analysis allow us to do? It allows us to make sure our sample size is big enough to answer the question we're asking. But it requires that we precisely specify the question, we identify the statistical testing method, and we identify the risk that we're willing to tolerate in type 2 and type 1 errors. Again, you rarely see this happen in diagnostic pathology studies, although it invariably happens in clinical trials for drugs. Now, how do you make the comparison? Well, some comparisons are better than others. Overall percent agreement, you see this a lot. It's generally a terrible choice because it depends upon the prevalence of the condition in the population. A test that's 1% sensitive for a condition that's 1% prevalent in the population has 99% overall agreement. Impressive, but meaningless. Or Cohen's Kappa. This is a measure of agreement that's, again, highly dependent on the prevalence of a condition in the population. So it can be a pretty good measure of how often pathologists agree on how to interpret a slide, but it's not very good at measuring how accurate they are in achieving a diagnosis. Sensitivity and specificity. Here we're getting better, and this is what we normally see reported. It's not sensitive to the prevalence within the population, and hence it can be used with population prevalence to compute positive and negative predictive value. And there are nice tools available to help you do this, do these calculations once you already have the data, and do confidence intervals. A nice set of tools are found in the clinical calculators on fasterstats.net. So we'll consider here two different tests, one of which here has a 90% sensitivity, 90% specificity, the next of which has a 90% sensitivity and a 95% specificity. And we put them up here and ask, okay, this is the data that we got. Do these really have different performance characteristics? Well, I used the 200 cases that I employed in those previous two slides, and you can see there's tremendous overlap in confidence intervals. So you can guess that you're probably not very safe in concluding that test 2 has a specificity that's higher than test 1. Okay, what does it take to cut the confidence interval down? Obviously, more cases will help. So to give you some sense of how hard it is to drive confidence intervals when you're up at high sensitivity and specificity, as you are with SARS-CoV-2 testing, consider this assessment, this simulation for test 2 at 200, 400, and 800 cases. And we see that the 95% confidence interval for sensitivity does shrink. It shrinks by almost 50% in going from 200 to 800 cases, an immense amount of work. And the confidence interval for specificity shrinks a little bit, but it doesn't shrink very much at the high end. There's very little room to work with when you're talking about highly specific tests. And this is a real problem then if you're trying to do comparisons of tests that are really, really good, such as, for example, all of the RT-PCR, or almost all of the RT-PCR tests for SARS-CoV-2. So now when you look down at test 1 at the bottom here and compare it with test 2, again on 800 cases, there is still overlap in confidence intervals. That's pretty difficult to work with. So there is a test, there are two tests that are commonly used to compare these two, McNamara's test and Little's test. They are based on paired comparisons. So the kind of study we frequently see with SARS-CoV-2 where we take one patient and we take two specimens and we compare the two tests or the two sampling techniques. You'll also see people try to use Fisher's exacta and chi-squared. These are inappropriate for paired comparisons. Paired comparisons are always correlated. These tests are good for uncorrelated testing. So they should not be used for these split sample types of tests that we're typically evaluating with SARS-CoV-2. So let's take a real example. These are cases reported by the Washington State Department of Health, and they have compared oropharyngeal samples with nasopharyngeal samples and done a sensitivity comparison. And they found down here nasopharyngeal samples to be apparently, perhaps, just a little bit better than oral pharyngeal samples. So we tested that with a two-tailed McNamara's test. And in fact, let me go back, sorry. We found a p equals 0.065. So now this gets you into the question of p-value. If you are simply testing the hypothesis, you would have rejected the hypothesis that these were different because our p was greater than 0.05. On the other hand, 0.05 is a magic number. The odds are still overwhelming. They're 16-1 that your nasopharyngeal samples are better than your oral pharyngeal samples in this case. So think about very carefully how you interpret these, this type of testing. We'll consider another test to talk about a challenge of calculating sensitivity and specificity. On the left-hand side, you'll see data. This was provided by the NCI FDA evaluation of Euroimmune serology. And they provide the raw data for this. They give an estimate of sensitivity and specificity together with their confidence intervals. Now, FDA makes a decision, and it's a fine decision, it's a correct decision, that for sensitivity and specificity calculations, equivocal results are counted as false negative if it's a positive sample, and a negative sample, an equivocal result, is counted as a false positive result. But that may not be the way you actually use the test in the laboratory. It may be that you're going to be discarding these equivocal results and just doing a different test entirely. Is this really a false negative? Well, I'll leave that up to you to decide, but I'll simply say that if you don't count equivocal cases, the sensitivity changes dramatically. It moves up to 96% and looks substantially better. Not enough better to overcome the confidence interval problem. This is a small study, but it does talk about a challenge. Okay, so suppose we were going to do this study of whether nasopharyngeal swabs were really better than nasal swabs, and this will be talked about this afternoon. And we're going to, I'm making this up, but let's just say that this is the way we're going to do this. I decide what my power is going to be, what my type 1 error tolerance is. I'm seeing positives in about 10% of the samples, so that'll give me an idea of what I want to do. I think there's going to be strong correlation because I'm doing safe sample. I'll need to use McNamara's test or Little's test. I don't have a statistician to help me. I'm a terrible programmer, so I'm going to use a calculator that's online. This is some data that Po2 published in the New England Journal. And we can see that the power associated with this particular data to tell a difference between nasal and nasopharyngeal swabs is actually pretty small. You would have needed about 5,000 specimens to show that they weren't equivalent with certainty. But the reason I take this data is to talk about biased and unbiased estimates. If you use the NP swab as a gold standard, and thus you assume the sensitivity and specificity is 100%, then the nasal swab looks like it has a sensitivity of 94% and a specificity of 99%, that you've just created false positives by using a nasal swab. That doesn't quite make sense to me. So suppose we consider a composite gold standard. And the composite gold standard in this case, it says if it's positive with either swab, then we will consider it a true positive. And we will assume that both are 100% specific. Well, in that case, your nasopharyngeal swab looks just a little bit less. It looks like it gives you a sensitivity of 98%. The nasal swab looks about the same, but we've just shrunk the difference by doing an unbiased or a less biased estimate. I won't call it unbiased, but I will call it a less biased estimate. Okay, so that's an awful lot of swabs I need to do this. I said I need 5,000 swabs. What if I just tested a bunch of patients and saw who ended up with the most positive. So an unpaired test. I do a sample size calculation for that, and I find I need 60,000 patients. That doesn't sound too terribly good. So the design based on paired specimens is always going to be more efficient. So let's talk about some lessons learned. First, it's very hard to compare two strategies or two tests that are both very good. Second, it's always better to use a paired sample strategy than an unpaired strategy if you can. Third, it's almost impossible to compare sensitivity and specificities for tests that are performed under very low prevalence conditions. Just the sample numbers get too high. Fourth, it's very easy to build bias into a test, and there's great temptation to do that when it's too laborious to create an unbiased ascertainment strategy. 5, it may be more useful to compare analytical performance of two tests than to compare clinical sensitivity and specificity. If you really care, you are likely to pick up something by having an analytically superior test, but you may never be able to detect that using clinical comparisons. Finally, a reminder that the p-value tells you nothing about relative clinical performance, only about the probability that the performance is different. It says nothing about how much different that is. And in fact, understanding how much two tests differ requires enormous sample sizes, and it almost never actually occurs even in clinical trials. I recommend CYS before you do these studies. Consult your statistician. A thorough statistical consultation will help you avoid failing to define the question properly, failing to choose a good gold standard, failing to identify potential sources of bias, failing to do the statistics properly, and failure to present the results in a complete and unbiased manner. And in 10 or 12 years of clinical trials that I did while I was at VA, these were probably the most important lessons I ever learned. I'd like to acknowledge an awful lot of people who provided input into some of the data that was here and into the discussions, and I will end.
Great. Dr. O'Leary, thank you so much for giving us a really excellent summary of the sort of statistical background of how to approach diagnostics. And in the interest of our overall program time, we're actually going to move forward with our next speaker.